Now the next question is, describe the formation of recombinant DNA by the action of EcoR1, and this question is of 3 marks. So as you know that EcoR1 is a type of restriction endonuclease that gives us sticky ends. So once we have isolated the DNA from vector and the source cell, then 2 DNA molecules are cut with the help of restriction endonuclease EcoR1 that actually cuts both DNA strands at the same site, that is between bases G and A of the palindromic sequence GAATTC. So when this restriction enzyme will cleave the DNA strands between G and A, we will get the sticky ends. Sticky ends because these are unjoined bases. Now these sticky ends, they will get ligated with the help of enzyme DNA ligase, and that results in the formation of recombinant DNA. And why are we calling this recombinant DNA? Because we are recombining or rejoining the 2 DNA strands from different sources or different genomes. So let's see the overview of this process that I just explained. So restriction enzyme EcoR1, it cleaves the foreign and vector DNA at the palindromic site GAATTC between the same 2 bases on the opposite strands. And this restriction cleavage leaves single stranded portions which are called sticky ends on each strand, and now these sticky ends are ligated with the help of DNA ligase to form recombinant DNA. So students, you can get 2 marks to explain this process in a diagram. And 1 mark for the explanation of this process. So this question can give you 3 out of 3 if you are explaining this process with the help of diagram and then explaining the steps of this process. Now this question has another option too, so let's see the another option of this question. So here is the next part of this question. Describe the process of amplification of gene of interest using PCR technique. So the amplification of gene of interest using PCR involves 3 steps actually. First, denaturation at 94 degrees Celsius, second, annealing at 50 to 60 degrees Celsius, and third, extension or you can say polymerization at 72 degrees Celsius. So out of 3, you will get 1 mark to explain these 3 steps. And next is the diagrammatic representation of the process of PCR explaining these 3 steps. So here you can see the first step, denaturation, where the high temperature breaks the hydrogen bonds between the 2 strands of original double-stranded DNA and will give us 2 copies of single-stranded DNA. Now the next step is annealing. Annealing is actually the ligation of 2 sets of primers with the single-stranded DNA templates. What is a primer? Primer is a single-stranded DNA or RNA that acts as the site of starting of DNA replication. So primer gets attached to the 3' end of the single-stranded DNA. Now the last step in PCR is extension, which requires the presence of a thermostable polymerase, that is Taq polymerase, as this step occurs at 72 degrees Celsius, and along with Taq polymerase that has been obtained from Thermus aquaticus, a thermophilic bacterium, we need deoxynucleotides that will act as a raw material for the extension of the DNA by polymerase. So this is how after these 3 steps we will get 2 copies of the DNA. So Earlier we had one copy of this DNA, now we have multiplied it, and after repeating this cycle for approximate 30 times, we'll get 1 billion copies of the desired DNA molecule. So for this particular step, you will get 2 marks to explain the diagram and the steps in detail. So this is how you will get 3 out of 3 after explaining the technique in detail. Now let's see the next question.